During class today, we started talking about neurons and how signals can be sent along a neuron. And it's pretty clear to me by the end of class, while you were understanding many of the different topics we were covering, there was definitely some confusion about action potentials, resting potential, where it all fits, kind of the order, where is this happening within the neuron. So I want to touch on a couple points that I hope will clarify some of the things we talked about today. So to begin with, remember we have a neuron cell. And the neuron cell has the dendrites, the soma, or also called the cell body, the axon, that long portion of the cell that goes to the end, and finally the axon terminals. The way a neuron works is the dendrites will receive signals or inputs from other cells. They might be from other neurons, they might be from other cells in general, but signals are constantly coming in here. Those signals are then processed here in the soma, and this is where it is looking at all the signals and trying to figure out what is background noise that can be ignored and what needs to be responded to because it's actually a signal being passed on. So the soma integrates and determines that. And what the soma is looking for is, do we cross that minus 50 millivolt threshold? If we don't, the soma does nothing. If we do, the soma is going to activate an action potential to take the signal down the axon. Remember, the axon can be a very long distance. It could be up to 3 feet long, the length of your arm and back. So we need a signal that can pass along there. So the soma will trigger an action potential, and that action potential will happen right here. And then it will cause another action potential to happen right here, and another one, and another one, and another one, all the way down the axon until we get to the axon terminals, at which point a message will be sent from this neuron to the next cell. That might be another neuron, it might be something else. So running through the neuron signals step by step, the first thing we have to do is to establish a resting potential of minus 70 millivolts inside the neuron. That is done by using the sodium-potassium pump, which moves 3 sodiums out of the cell and 2 potassiums into the cell. So we are creating not only a concentration gradient of molecules, but also an electrical gradient. That outside the cell is more positive, while inside the cell is, relatively speaking, more negative, fewer pluses. Potassium is also leaking out due to the concentration gradient and being pulled back in by the electrical gradient. And the balance is reached at around minus 70 millivolts. So that's where the cell is in balance, and it will maintain that as long as nothing else happens. At this point, the neuron will begin to receive various stimuli, also called signals, along the dendrites from other cells. It will receive hundreds, if not thousands of these. Each stimulus or signal will cause a small number of sodium ion channels to open. This in turn will make the inside of the neuron more positive. The question then becomes, how much more positive? Very small signals, small stimulus, will make it more positive, but it will not reach the threshold value of minus 50 millivolts. So it will not reach that. In that case, nothing happens. It'll go up a little bit more positive, then drop back down to our minus 70. However, sometimes enough stimulus is reached that the cell's threshold hits minus 50 millivolts. So it goes from minus 70 to minus 50. When that happens, the soma will trigger an action potential along the axon. The action potential is a signal that travels down the length of the axon. It will be a series of action potentials. So not just one, it's multiple, one right after the other, that will travel from the soma down to the axon terminals. Finally, once the axon potential or the action potential reaches the axon terminals, it will activate the next process, which we call synapse, where we pass the signal from one cell to the next. Synapse is going to be our focus in this lecture. We haven't talked about synapse yet. My hope is this is giving a little better idea of kind of the step-by-step what's happening. The last thing I want to go over, reviewing from today's lecture, is action potential itself, what's going on. So this top diagram here, we have, this is the resting potential. Okay, so we have our resting potential. And if you remember right, we have lots of pluses on the outside of the cell and fewer pluses on the inside. The whole thing means inside the cell is minus 70 millivolts compared to outside. There's more pluses on the outside than inside. We also have a lot of sodium on the outside and a lot of potassium on the inside. And we're sitting there, minus 70, waiting for something to happen. We have now received enough signals that we have hit threshold. So the inside has gone from minus 70 to minus 50. And an action potential is going to be triggered. When the action potential is triggered, so we hit the minus 50 right about here. When the action potential is triggered, the sodium ion channels in that area at the beginning of the axon all open up. And sodium rapidly flows into the cell because of the concentration gradient. What we are doing, we are taking many pluses from outside the cell and moving them into the cell. That is going to make the inside of the cell more positive. So if we're looking at the value inside the cell, we're going from about minus 50 up rapidly all the way up to about plus 40. We call this depolarization. Okay. Depolarization continues until we get to plus 40. At that point, something else happens. The sodium channels close and the potassium channels open. So now the potassium channels open. The potassium, which is also pluses, is now flowing rapidly out of the cell. So we are removing pluses from inside the cell and putting them outside. That is going to cause our inside our cell to go from a positive 40 rapidly down to negative 80, maybe even negative 90. And we call this repolarization. So we've reached this repolarization down to minus 80, maybe minus 90. So we passed our normal resting potential. At this point, everything closes. And the sodium-potassium pump takes over, combined with some potassium leaking through, and we reestablish our resting potential at right around minus 70. That is how an action potential occurs. That's the details of each step. The action potential is a series of steps of different messages being sent down the lake of the axon until it reaches the axon terminals. So now, let's talk about what happens when it reaches the axon terminals. So now we're talking about the nervous system, and we're going to incorporate a chemical messenger called neurotransmitters. The process of passing a neural message from one cell to another cell is called neural synapse, or often just synapse for short. To understand this, we need to define a couple terms. First off, we are involved two cells, the cell sending the signal and the cell receiving the signal. The presynaptic cell is the cell that is sending the signal, before the synapse. The postsynaptic cell is the cell that is receiving the signal. And then finally, we have what's called the synaptic cleft. The synaptic cleft is the space between the two cells. They're not physically touching each other. There is a small gap 20 to 30 nanometers, so tiny, tiny amounts, but it's a gap. So the presynaptic cell is going to release a message that is going to cross the postsynaptic cell and be received, it will cross the postsynaptic cleft and be received by the postsynaptic cell. That is neural synapse. And when we do this, we have two different types of messengers we can use. The first one we can use are chemical messengers called neurotransmitters. Or we can use electrical currents as the synapse, and that will be an electrical synapse. So we're going to go through each of these, but let's talk about chemical synapse because they are by far the most common type. So a chemical synapse, it occurs when a signal is being sent from one neuron to another cell. Often, this is one neuron sending to another neuron. However, in this example, we're going to talk about a neuron, a motor neuron, sending a message to a muscle cell, or a group of muscle cells, telling them to contract. So this would be a neuromuscular junction, where a neuron, nerve cell, and a muscular cell come together. And if you look at our diagram here, we have our neuron. Remember, the message will be sent in this direction. And at the end, the axon terminals are connected to muscle cells, these muscle fibers. So what we want to do, we want to pass, the action potential is going to go down the axon, and it's going to get here, and then when it reaches the axon terminal, we need to use a chemical messenger to cross over that synaptic cleft and trigger the muscles and tell them it's time to contract. So how are we going to do that? Well, if I zoom in here and I'm looking at where the axon terminals touch the muscles, you'll see I have these kind of bulbs or round puffy areas at the end. Okay, these are called presynaptic terminal buttons. Okay, so these buttons are where the interaction between the neuron and the muscle cell is going to happen. And you can see right here a little rectangle. That rectangle is going to be the next picture. So here we go. This is what's inside the rectangle. Up here, we have the neuron. So this is the neuron. And then down here, we have the muscle. I could just as easily say that we have one neuron passing to the other neuron. The neuron in this case, up here, is the presynaptic cell. And down here, the muscle is the postsynaptic cell. So the neuron will be sending the signal, the muscle will be receiving the signal. So let's walk through the steps of what's going to happen for a signal to occur. Okay, so our presynaptic cell has been sending an action potential down the axon. So here it comes, action potential, coming down, and eventually it reaches the end. It reaches the axon terminal. So what's actually going to happen here? Okay, when it reaches the end, we are going to have elevated levels of sodium inside the cell because the action potential caused sodium to enter the cell. What that sodium is going to do is it is going to open up some ion channels. It's going to open up, so this sodium will go here and will open the ion channels. These are very particular ion channels. What they are going to do, they are going to let in calcium, Ca2+, flowing into the presynaptic cell. So we now have calcium 2+ moving into the cell. Once the calcium 2+ moves into the cell, what is going to happen is the calcium is going to cause these vesicles, which contain a neurotransmitter called acetylcholine. Okay? So our neurotransmitter is called acetylcholine, sometimes abbreviated ACH. Acetylcholine is the neurotransmitter that stimulates muscle contraction. Okay, if we were triggering another cell, not a muscle cell, we would use a different neurotransmitter. But these vesicles are sitting here containing the different neurotransmitters that I might use. When the calcium moves in here, it causes these vesicles to form and bond with the cell membrane, releasing the neurotransmitters into the synaptic cleft. So you can see each of these dots is acetylcholine. It is now going into the synaptic cleft, at which point it will start to spread out through diffusion. We have a high concentration here. It's going to spread out. Some of those acetylcholines will go across, and there are receptor proteins on the other end. And you can see the acetylcholine went in here. It's acting as a ligand, and it is opening the ion channels. These ion channels are going to allow sodium and potassium to flow in and out of the cell. Sodium's moving in, potassium's moving out. This is going to cause depolarization, which in turn is going to trigger an action potential in the muscle, which is going to cause the muscle to contract. The final step here is the acetylcholine ligand that is attached to the ion channel is going to release and separate from the ion channel, closing the ion channel back down. Okay? So, running through those steps one more time. The first step, action potential travels down the axon and arrives at the axon terminal. We now want to send the message to the postsynaptic cell. When the action potential arrived down here, it provided a lot of elevated sodium in the cell because the action potential allows sodium to move in. That sodium triggers the opening of calcium ion channels, which allow calcium to move into the cell. The calcium causes vesicles filled with our neurotransmitter, acetylcholine in this case, to bond with the cell membrane and be released into the synaptic cleft. The neurotransmitters spread by diffusion, and some of them find their way over and bond to ion channels in the postsynaptic cell. These ion channels then open up, allowing sodium to flow in, potassium to flow out, and cause depolarization. This in turn triggers an action potential in the muscle cell, which results in contraction of the muscle. Finally, the ligand, acetylcholine, releases from the ion channel and closes the gate, ending the message being sent. That is how a message is sent from a presynaptic cell to a postsynaptic cell using chemical signals like neurotransmitters. So what happens to the neurotransmitter after the signal passes? Well, we want to get rid of that neurotransmitter as quickly as possible. We don't want to leave it in there because it can keep triggering ion channels over and over after we're done. So we need to clear them away. And your body does that in a couple ways. First off, diffusion is going to take care of some of it. Some of them are just going to leave the cleft and be gone. They're not going to be in the area, so they don't matter. Some of them will be recaptured and put back inside the neuron to be used again in the future. And finally, there are enzymes out there that will help break down any remaining ones so they don't stick around and mess the system up. Blocking the removal of neurotransmitters actually can cause tremendous problems. I don't know if you've ever heard of sarin gas, but sarin gas is a type of nerve gas. It was used as a chemical weapon for a long time. It has now been outlawed. But sarin gas, the way it works, it causes paralysis and death simply because it inhibits the enzymes that break down the neurotransmitters. So people who are exposed to sarin gas their muscles would contract and they would continue contracting over and over and over and stay locked because the neurotransmitters would stick around, causing those ion channels to remain open. And so the person wouldn't be able to relax the muscles, and it would result in seizures, paralysis, and ultimately death. So getting rid of those neurotransmitters, definitely important. So far, we've talked about chemical synapse, how chemical messengers, neurotransmitters, can be used to pass the message from a neuron to another neuron, to a muscle cell, or some other cell. But that's not the only kind of synapse. Another kind is what we call electrical synapse. Electrical synapse rely on the movement of electrical currents to pass the message from one cell to the other instead of neurotransmitters. So basically, they are simply continuing the action potential from one cell into the next. And that is possible because what the currents do, rather than having a true gap between the cells are bonded together using gap junctions. Think back, we talked about gap junctions a long time ago when we did the cytoskeleton. Gap junctions can connect different cells together and let ions and other things flow directly from one cell to the other. Well, if we have gap junctions between two cells, when an action potential travels down our presynaptic cell, the sodium will flow right through and continue the action potential down the next cell. This is an incredibly rapid, swift way to pass messages. It's faster than neurotransmitters. But it's kind of an all or nothing. There's no way to modify it at all. You can't release different neurotransmitters and so only trigger some cells but not others. You can only continue the action potential down it. So this is a good system for messages that cause a very rapid and unchanging or unvarying neural pathway, something that is always done the exact same way. And a great example of that is when like a giant squid or a lobster needs to escape danger. What a lobster will do, you look at my hand here, a lobster has a tail like this, and lobster will frequently hang out underneath rocks. And if a predator wants to get it, the predator will come in toward the head and try to grab the lobster. Well, the defense the lobster has, some lobsters have these giant pinchers, but many don't. And so what the lobster will do, it will very rapidly flip its tail, which will yank it back underneath the rock out of the way of the predator. And that is always done the exact same way, full strength, jump backwards as fast as you can. So that is a good behavior that can be controlled by electrical synapse. But other behaviors that we might want to modify in different ways, or I might want to send a signal down a neuron, but I want to have some control over how big of an impact it has. I don't want to contract all my muscles. I want to contract a few, so I have a light touch or a very strong touch by contracting more. That flexibility is more easy to obtain using chemical synapse, neurotransmitters, than electrical synapse. So that's the reason you'll see both types out there. Chemical synapse are definitely more common, but electrical synapse do exist. Okay, let's go back to the receptor side. So we have a postsynaptic cell, it's got a receptor there, and we actually have two different types of receptors. The first one we've already talked about. Ionotropic receptors, basically ion channels. Okay, ligand-gated ion channels, like the acetylcholine receptor we already talked about in the muscles. These are very common types of receptors. The neurotransmitter travels across the synapse, opens up the ion channel, ions can flow in or out of the cell, passing a signal along. This is great for fairly fast messages that we want to send, short-lived changes, so I want a very quick response. It's not going to last for long, I just want it to do something quickly, and I want an immediate change, like contract a muscle. Okay, ionotropic receptors are great for that. But the other kind of receptor is a metabotropic receptor. These are not ion channels, so no ion channels. Instead, these are usually G protein linked receptors. So rather than opening an ion channel, there will be a receptor protein that will receive the neurotransmitter, and it will then activate a G protein that will then cause a cascading event, just like cell signaling, what we've talked about before. The advantage of this method, it's slower, but because the cascading event can do many, many different things, you have a very wide range of effects that can happen, even going so far as gene expression. By messing around with different transcription factors, we can turn genes on or off using this method. So metabotrophic receptors are generally used for slower, very long-lived effects that I want to activate. Development, growth, these are the kinds of things that might be involved there. But ionotropic for very quick results that I need immediate responses. So both of these receptors exist. They work along similar ideas. It's just the responses they give. Do they open an ion channel, or do they cause a cascading event that will have some effect in the cell? Finally, what I want to talk about here is signals. Okay, we have been talking about pre- and postsynaptic cells from the sense of the presynaptic cell is sending a signal to the postsynaptic cell. But now, let's talk about the postsynaptic cell as receiving the signal and what it's going to do. When a postsynaptic cell receives the signal, this is basically our neuron receiving a signal at the dendrites. The dendrites are the postsynaptic part of the cell that are receiving the signal. And as we said earlier, not all signals are going to cause an action potential. So why is that? Well, when a dendrite receives, or a postsynaptic cell receives a signal, one of two things can happen. Many of the signals are going to be excitatory postsynaptic potentials. They are excitatory. In other words, they're going to open up a ligand-gated channel that is permeable to potassium and sodium. This, in turn, is going to depolarize the cell, moving the cell's potential toward threshold, minus 50 millivolts. Okay? If it reaches minus 50 millivolts, action potential. If it doesn't reach minus 50 millivolts, it will return to minus 70 gradually. But there are other signals that are received that are what we call inhibitory postsynaptic signals, or potential, IPSPs. Inhibitory ones are ligand-gated ion channels, but they don't let sodium potassium in. They just let potassium out and Cl minus chloride in. When they do that, they hyperpolarize the cell. They make the cell become more negative. This moves the membrane further from threshold, making an action potential less likely. Now take a look at this picture. This is a picture of a neuron inside a mouse's brain. And every single one of those little yellow dots you see is a signal being received by this neuron. Those are the dendrites sticking out from the neuron. So dendrites do not receive like one signal. They are receiving hundreds or thousands of signals at any given moment. Why do we get some of them triggering, some of them don't? Well, we have a process called summation. In general, a single synapse, so a single neurotransmitter coming across and opening up a sodium channel, is not enough to cause an action potential. But we have summation. Multiple signals combined together can cause or trigger an action potential by reaching the threshold of minus 50 millivolts. So you might have two excitatory potentials that together can go ahead and cause us to reach the minus 50 value. Or you might have two that don't reach that. So multiple synapses can sometimes result through summation in triggering an action potential. At the same time, we also have inhibitory potentials that are moving us away from having a action potential occur. Okay, so let's look at how this can play out. Here I have a neuron, and you can see different excitatory or inhibitory signals are coming in. In this first graph, we get an excitatory signal, then a pause, then another excitatory signal at the same location. And here's what happens. When my first excitatory signal comes in, some sodium channels open and it bounces upwards, but it does not reach threshold. And then nothing happens, so the sodium-potassium pump gradually brings it back down to -70. And then another excitatory signal comes in, pops us up, but does not reach threshold. And so our cells are never going to respond and create an action potential from that. It treats that as background noise we can ignore. Now, summation. I'm going to receive two signals in the same spot, one right after the other, but this time, my first signal has occurred, has not gotten me to threshold, but the second signal happens before the first one is returned to resting potential. That one bumps up enough that we hit threshold, -50, so it causes an action potential. Okay. Let's look at this third one. Here we have two different excitatory signals coming in at different locations. So one here at E1, a different one at E2 at the same time. Because it's two of them, they have a summative effect. They bump up more than either one does by individually, getting us to -50 threshold action potential will occur. What is normally happening in a cell is you are constantly getting excitatory signals and inhibitory signals. So you might get an excitatory signal that does not quite take it up to threshold, then an inhibitory signal which drops us down to like -80. You might get excitatory and inhibitory at the same time, which almost cancel each other out, or different combinations. So the thing to remember is an action potential requires us to get to -50 to hit threshold. If we do that through summation, multiple signals coming in, we will get an action potential. If we don't reach that threshold, -50, no action potential will occur, and the signal will not be passed down the neuron. There are many types of neurotransmitters. Okay, remember, signals depend on the neurotransmitter being released to another cell, but what that cell does, or what cells will respond, depend entirely on what kind of receptors occur on that postsynaptic cell. Sometimes they are ligand-gated channels and they're just going to pass an action potential on or contract a muscle. Other times they are G-protein coupled receptors, metabotropic receptors, that instead of continuing an action potential will cause a cascading event inside the cell, like normal cell signaling, and will cause some often long-term effect. Neurotransmitters, we've already mentioned acetylcholine is a common neurotransmitter for muscle contraction. Many amino acids will act as neurotransmitters, glutamine, glycine. We can have biogenetic amines like dopamine, serotonin, norepinephrine. These are things that are also neurotransmitters. Neuropeptides, so different proteins can act as neurotransmitters. Some gases can act as neurotransmitters. Nitric acid, nitric oxide, not acid. This is laughing gas. This is a neurotransmitter. It numbs your nerves, and that's why they use it for pain in like dental procedures and stuff. It's an anesthetic. Okay, you do not need to memorize all the different neurotransmitters out there, but I want you to be aware there are a wide range of neurotransmitters, and what each of them does depends entirely on what kind of receptors are in the postsynaptic cell and what those receptors do. Okay, our final topic for today is what we call synaptic plasticity. Have you ever started to learn something new? Maybe playing a musical instrument or taking up a new sport or activity, hobby. When you started doing it, it was really hard to do, wasn't it? I don't play the piano, but I can only imagine playing the piano when you start learning how to press the different keys and then learning how both the hands do different things. That does not come easy. A tremendous amount of work goes into learning to do that. And we tend to think of it like, oh, well, I learned how to do it, and that's why. But what does that actually mean? What scientists have discovered is that repeated use of neural pathways can change those neural pathways. When you do certain motions or use certain neural pathways over and over and over, you get better at them. The pathways improve. The neurons will physically grow larger. Okay? The synapses will strengthen. The number of postsynaptic receptors will increase. That cell, the postsynaptic cell says, I get this signal all the time. It's important. Let me put more receptors out there so I can respond that much better to it. Also, we can start getting more neurotransmitters stored. We can start getting more secondary messengers if it's a G protein receptor. More responses prepared because your body is learning. This is something we do a lot. And as your body adapts in this way, you get better at the activity. Suddenly, you can play the piano without even thinking about it. Your hands just kind of know where to go. What used to be hard of throwing a football to someone standing a far distance and making sure it reaches them is now possible without really even thinking about it. The more you practice something, your neural pathways get reinforced, grow, and get better at carrying those messages. We call this learning or memory. By the same token, if you stop using neural pathways, they weaken. They start to atrophy. The neurons get smaller. We start removing some of those postsynaptic receptors. We're going to use those proteins for other things. Fewer neurotransmitters, fewer secondary messengers, and so on. This is why practicing can make you better, and not practicing does not make you better. It can make you worse. You can lose skills. Your brain and neurons, your nervous system, is literally changing to get better at the activities you do repeatedly. And it is forgetting or getting worse at activities you don't do often. This is why you get me saying things like, study every day for the AP test. We've got 29 days to go. Sometime, little each day, you will grow those neural pathways. If you try cramming in over a 2-3 day, maybe even a 1 week period, you're not going to develop those neural pathways nearly to the extent, and you won't learn as much. It won't go in. So there's science behind it. Practice really does reinforce and make you better. Hey, that is the nervous system. Hope it helped. Hope you clarified a little bit from today's lecture, and now you understand how neural synapse work. If you're confused on anything, make sure to bring those questions with you to class, and we'll go over it. Good luck, and I'll see you in class.